Hi, my name is John Negrete. In this video, I will be presenting the current limitations in ClinVar and will also provide you with a tour of ClinVar entries. This video was recorded in October of 2023. I have been an intern at the Center for Genomic Interpretation since June of 2023. CGI was founded in 2017 and is an independent 501c3 nonprofit organization with the mission to save and improve lives through encouraging careful stewardship of clinical genetics, genomics, and precision medicine. The purpose of this video is to inform users of ClinVar about the strengths and limitations of different information found in ClinVar entries for germline clinical genetic variants. By the time you get to the end of this video, you should feel confident with knowing what information to trust and what information to investigate from within ClinVar. Note that ClinVar updates frequently, changing its interface, so this video will become out of date in time. The ClinVar website already has an excellent help section with tutorials on how to search for variants in ClinVar. I will be putting that and other links in the description below. I won't duplicate the explanation here, but if you're new to ClinVar, be sure to review those tutorial pages. As I explore ClinVar with you in this video, we will be taking a look at 3 germline variants in ClinVar as examples. First, the most well-understood pathogenic variant is cystic fibrosis. Then one of the sickle cell variants in HBV that confers resistance to malaria. And lastly, a rare silent variant in the MYH7 gene. Pathogenic variants in MYH7 can lead to hypertrophic cardiomyopathy, although this particular variant we'll be talking about probably does not. Variant identifiers and other data that are available about a variant or condition from a public resource are added by ClinVar and NCBI staff. NCBI, or the National Center for Biotechnology Information, is a government organization under the National Institute of Health that provides online resources for molecular and genetic information and data, such as PubMed. These staff may also add published allele names, citations, and linkouts. While infrequent, these entries direct from ClinVar or NCBI staff could contain error, so always be mindful of this possibility. If you come across an error you recognize, it is helpful to let the ClinVar staff know so they can investigate it and possibly fix it. ClinVar has a review status for each variant page, ranging from 1 to 4 stars, that is automatically assigned by ClinVar in an attempt to communicate the trustworthiness of any variant classification assertion. Conflicting information is not surprising for less studied and encountered variants. And even with more studied variants that show there are no conflicts, we can still find some meaningful differences in ClinVar submissions from different labs once you dig further into the entries. As a disclaimer, ClinVar states that they do not curate or modify classifications of submissions from labs and researchers, and that they provide only limited curation of variant and phenotype terms. This means that these parts of any ClinVar page could be in error. The information in this top blue section of a ClinVar variant entry can be seen as the general housekeeping information for a variant. Unlike the CFTR and HBB variants, this MYH7 variant is not as well represented in ClinVar. Currently it has only 3 submissions, compared to the 72 submissions for the CFTR variant and 54 for the HBB variant at this time. Note that ClinVar's submission from labs, researchers, and others is voluntary, and not all labs contribute all of their variants into ClinVar. It's not uncommon for clinical labs to submit just enough variants into ClinVar for them to meet the expectations of the clinical community, while strategically holding back other variant classifications for a variety of different reasons. We first see in these top blue sections a variant represented by its standardized HGBS nomenclature, consisting of an NM transcript, the gene symbol, the coding DNA reference sequence, or the C dot, the nucleotide change, and the protein change, if any. We then see the consensus classification of the variant, which in this case is likely benign. This section is a result of all the interpretations from various submitters. Keep in mind that for variants that are not well characterized, meaning there haven't been a lot of studies done on the clinical impact of a variant, the consensus classification may be biologically and clinically wrong, even when 20 or more ClinVar submitters might be in agreement. If the labs are all using the same limited and incomplete data, with roughly the same variant classification protocols that are probably based on a generally accepted guideline, Many different labs can arrive at the same variant classification conclusion, which may not be biologically and clinically true. This was demonstrated for several hereditary cancer variants in this 2015 publication that showed why several positively classified variants had to be downgraded. There will be many more such downgrades in the coming years across many genes, as peer-reviewed literature has been rolling out over the last several years that is demonstrating that ClinVar has many false positive classifications. More peer-reviewed literature keeps demonstrating that ClinVar has many false positive classifications, even for consensus classifications. Importantly, for genes that have loss-of-function disease mechanisms, consensus but false positive classifications are likely most common for truncating variants in parts of genes that have alternatively spliced RNA transcripts that rescue the protein function, but those rescuing transcripts haven't been characterized yet by the scientific or clinical community. In the coming years, as genes get better characterized for the clinical context, even though they're already being used in the clinical context, there are going to be many downgrades of variants from pathogenic or likely pathogenic to variant of uncertain significance, likely benign, or even benign. Unfortunately, false positive classifications are more common in non-Western European ancestry patients, as was shown in this publication from 2016, mostly because the growing databases have more data on Western European ancestry patients, which allows for more accurate classifications in these populations. Also keep in mind that some lower quality clinical labs don't actually have a variant classification team at all, or at least not a very good one, and instead they may just take classifications already in ClinVar and echo them without their own informed reviews. Those labs may then submit their classification as if they've done an independent and well-informed review, while they haven't. In these cases, a variant receiving a 2-star rating in ClinVar indicating multiple submitters with no conflicts might actually not be as trustworthy as you would think, particularly if the first classification wasn't well done. This problem has been made even worse as some health insurance companies have required labs to submit their classifications into ClinVar, but the health insurance company doesn't have or enforce the requirement that the classifications should be independently reviewed by a high-quality variant classification team. Below the review status in ClinVar, we see the total number of submissions for this variant, followed by the first in ClinVar section, which is the date when the VCV, or variation ClinVar record, first became public in ClinVar, and then the date of the most recent submission. The last evaluated section contains the most recent date that the variant was interpreted by a submitter. Keep in mind that there is a lag of unknown time between the lab classifying the variant and then uploading their classification with ClinVar. And then there can be a delay of up to months before the ClinVar workers get it onto the live version of ClinVar. The accession section contains an entry beginning with the VCV, the number before the decimal point. This number is created by ClinVar to refer to all of the information from all submitted records interpreting the same variant or set of variants. Variation ID is a unique identifier assigned by ClinVar for the set of sequence changes that were interpreted. And lastly, the description section refers to the change that is being seen in the genome as a result of the variant. It is not intended to reflect the impact of the change on the gene product. For example, a single nucleotide variant doesn't sound like a big deal, but might have the possibility of completely abolishing gene expression. The example shown here is probably not doing anything like that. As we move down the ClinVar entry page, on the left you see the tabs for variant details, conditions, and genes. I'm going to come back to the variant details in a bit, but first I'll summarize these conditions and gene sections. The conditions sections provides a summary of the submitted interpretations for the variant, including the total number of submissions for a specific interpreted condition and interpretation combination, its review status, the date it was last evaluated, and a link to the reference ClinVar record. In this tab, we may also find a list of clinical features observed in individuals with this variant, such as here for our CFTR variant. In the gene section, we see a summary about each gene that is affected by the variant. We can see the gene that is affected, a link to an OMIM entry for the gene, the ClinGen gene dosage sensitivity curation scores, which if containing text that is hyperlinked, will take you to the ClinGen website for the dosage sensitivity summary of the gene, a variation viewer column that will link you to the NIH variation viewer page, and the related variants column, which contains the links to other variants from this gene in ClinVar. The link under Within Gene shows you variants that only affect this gene. which happen to be mostly short variants. The link under All shows you any variant that affects this gene, including both short variants and copy number variants. While most variants in ClinVar are within a single gene, copy number variants can affect more than one gene. In these instances, the gene tabs display the first 10 genes, with preference given to genes that are curated by the ClinGen dosage sensitivity map. ClinVar provides links to the NCBI's variation viewer, the UCSC browser, and the ClinGen dosage sensitivity map to view all the genes affected by a variant. Back to the variant details section, we once again see the variant represented by the HGBS nomenclature in combination with the gene symbol. The allele ID entry is an identifier for each individual nucleotide change in the set of variants that were interpreted. In other words, if the variation reports for interpretation of a haplotype defined by 5 single nucleotide polymorphisms, each single nucleotide polymorphism has its own allele ID. The set of a single nucleotide polymorphism that make up the haplotype are represented by the variation ID. The data in the variant details section may come from submitters, but may also be added by NCBI, especially when the variant can be mapped to the genome. According to ClinVar, all of the following sections beneath allele ID contain information that is usually provided by NCBI staff. Next, we see the variant type section. Our MYH7 variant is described as a single nucleotide variant. In the case of our CFTR variant, which is a 3 base pair deletion, the entry in that section is deletion. Next is variant length. Our MYH7 variant results in a single nucleotide change, hence the 1 base pair variant length entry. For our CFTR variant, this entry is 3 base pairs, since it is a 3 base pair deletion. The cytogenetic location section uses a standardized nomenclature to refer to the position of variants on a chromosome. The genomic location section provides 2 sets of coordinates based on 2 reference genomes, the GRCH38 and the GRCH37. The links to the side of these reference genomes take you to the genome browser either from NCBI or UCSC. Note that there are more complete and diverse reference genomes that have recently come from the National Institutes of Health, but at this time, those reference genomes are not validated for clinical use. The HGBS table contains various LRG, NG, NC, and NM reference sequence coordinates. More information about this section, such as main select transcript, can be found on the matched annotation from NCBI and EMBL-EBI webpage. I'll put this and other links in the description below on YouTube. The protein change section contains a predicted protein change that resulted from the variant mutation. In the case of our HBB gene, we will see that the entry is E7V, which is a glutamic acid to valine change at the 7th amino acid. Note that in some rare instances, the prediction may not match biology, as some unpredictable things can happen. The other name section contains the various other ways a variant is being or has been referenced in the literature. Please note that this section is rarely filled in, and if it is rarely exhaustive. If you're using this section to search legacy literature and you're wanting to be thorough, this section is likely not going to give you enough legacy naming of important variants. The canonical SPDI, or sequence position deletion insertion section, contains a notation that represents an observed variant sequence using deleted and inserted sequences at a given position in a reference sequence. In this case, we see the sequences NC_000014.9, the position as 234-13809, the deleted sequence, in this case only G, and the inserted sequence, in this case only A. Each section is separated by a colon. The functional consequence section contains information on the impact that this variant has, such as quantitative effects on gene expression, alternative splicing, and loss of function. This section is rarely complete or exhaustive, in part because it must be manually entered by a willing person, which rarely happens, but most often because the studies just haven't been done. Note that just because studies have been done, and something might be entered in here, does not mean that the entry is accurate, or the studies were done well. Keep in mind that most functional studies done on variants are of research grade, and the findings are unlikely to have achieved the necessary quality to be rightfully considered implementable in the clinical setting. The global minor allele frequency section is defined as the global minor allele frequency calculated by the 1000 Genomes Project. This allele frequency is for the most common variant at a position that deviates from the reference genome. It may not actually represent the allele frequency for a variant entry if the variant is not the most common variant at that position. Next, we see the allele frequency section, which can contain information from sources such as 1000 Genomes Project or GNOMAD. This may not be completed even if there is data in these resources. Also note that if you plan to rely on the data here, you'll need to go to the source to do your own quality check, like making sure there is sufficient sequencing read depth or population representation in a region to trust that a number is truly zero. Lastly, we see the link section, which contains links related to databases such as DB, SNP, and ClinGen. To better understand the limitations of the variant details section, let's compare a couple of our variants of interest. Because our MYH7 variant has not been reported by many labs, it is not too surprising that some of its data sections, such as other names, are empty. However, it can be surprising to see these sections are empty for some well-known variants. The CFTR variant, which results in the deletion of one amino acid, does not have anything entered into its protein change section. The protein is indeed changed, so ClinVar users need to be careful with how they interpret the absence of information in ClinVar. The absence of information does not necessarily imply that nothing is known. Another thing to be aware of is that the functional consequence sections might be empty even though there are functional consequences. Once again, the absence of information in ClinVar does not mean that there is not information. It's a hit or miss on whether many of these fields have had someone enter data in, and there could even be error or incompleteness in the entered data. For example, the HBB functional consequence field is empty, but this variant does have a well-understood consequence. The same field for the CFT variant shows only loss of function. Note though that there is much more detailed information about the functional consequence of this CFTR variant than what is shown in this ClinVar field and the included link. So be careful with what assumptions you make about this data field. Each of these variants has an associated global minor allele frequency. However, in the allele frequency section, separate from the GMAF section, CFTR has no information. This is curious since this variant can indeed be found on GNOMAD, which is one of the sources for information for the HBB gene allele frequency. So again, these fields may not always have the correct and complete data in them. We will now look at the submitted interpretations and evidence section. Here we will encounter different interpretations and associated conditions for the same variant by different submitters. Once we get down to this section, it is again really important to remember that these fields are voluntarily added by submitters and are not being quality controlled or assured by any ClinVar or NCBI workers. For well-characterized and well-understood variants, most of the entries are likely to be correct, but even here for these well-understood variants, it is not unusual to see entries of lower than desired quality. For example, with our CFTR variant, we can see that it has a total of 72 submissions of 4-star review status and is designated as a practice guideline. Despite the majority of submissions having seemingly been filled out properly, going through the entry we will see that there are 19 submissions that list this variant as pathogenic, but do not provide an associated condition. We will also see 2 submissions with an associated condition, but without any interpretation provided or any assertion criteria, and even 1 submission without any of these 3 sections filled out and without any clarifying comments. With our HBB variant, we see 2 submissions listing this variant as pathogenic for HBB-related disorders, 1 submission listing this variant as pathogenic for a condition listed as C cases without any further explanation, and 1 submission listing this variant as pathogenic for inborn genetic diseases. The reason this really matters is that for variants that are more rare and aren't well characterized, it is much more common for the 1 or 2 submitters for such variants to give incomplete and sloppy information in these fields. This next section is not present for all variants, and in this case is only found for our CFTR variant. Note that even when there is information in this section, it tends to lack nuance. This cystic fibrosis pathogenic variant, for example, has very well characterized functional consequences with lots of nuance and details that can be found in resources not linked out by ClinVar. The link that's actually in ClinVar under the functional consequence column takes you to the sequence ontology website, where we can see how they came to categorize our variant for a loss of function variant, but it does not provide much more detail. Near the bottom of the page is a section for citations for this variant. The take-home message here, before I get into the details about how these entries are structured in ClinVar, is that this is a poorly curated section. For many variants, there will be important citations missing from this section. Also, the citations listed here may not be fully relevant to the variant of interest. Please do not visually inspect this section for citation count and mistakenly interpret high citation counts to mean that the classification is therefore well-founded. The title of citations, author, journal, year, and the link information are provided here. This table includes all citations except for those submitted on the condition and citations submitted as assertion criteria. However, for the HBB gene, there are 17 entries that have no information other than the title. For some of the entries, the title is just listed as a PMID or a link. A few of the entries titled as a PMID are even actually repeats of other entered citations. The CFTR variant page has only 9 citation entries with only titles provided. However, the majority of them are only listed as links with no additional information. It is important to note that there is a good chance that the citations listed do not include the specific variant of interest. There may be literature on some of the associated conditions listed in the previous sections, on the gene in general, as well as on other variants at this genomic location, which may or may not be relevant to your understanding of the variant of interest. The next and last section is titled Text-Mine Citations, and it references our variant by its associated RSID. In the case for our HBB variant, it is RS''334. Clicking on the blue box titled LitVar2 will take you to a new page, the LitVar2 website, where you will see text that references this variant. Note that not all variants have this feature available in this section. Also note that this section states that these citations are identified by LitVar using the RS number, so they may include citations for more than one variant at this location. Please review the LitVar results carefully for your variant of interest. The citation list available here does not come close to being complete for most variants. So in summary, ClinVar is a useful research tool, but almost all the information found within it must be carefully and independently checked for accuracy and completeness if being used in the clinical setting. We are grateful to all those that submit high quality and more complete submissions to ClinVar. Please also remember that ClinVar updates frequently, so the information provided in this video will become out of date.